Okay, I'll just present it. Everybody else can see it as a full screen, is it?
Yes.
Okay, so the title of my talk is on improvements in strongyloides torcoralis serodiagnosis using new antibody and antigen detection assays. I'm Rahmah Nordin from the Institute for Research in Molecular Medicine, University of Science Malaysia in Penang, with my colleagues. Next slide, please. Okay, strongyloides torcoralis is a parasite that is present worldwide with more than 600 million infected in about 70 countries, in tropical and subtropical climate countries. The transmission of this parasite is by contact with contaminated soil, either directly or indirectly. The problem with this parasite is the phenomenon of autoinfection, which means the parasite can be maintained in humans almost lifelong. And upon immunosuppression, especially with treatment using corticosteroids, autoinfection may lead to hyper and disseminated infection, which can be highly fatal. Next slide, please. Okay, the laboratory diagnosis is traditionally done by microscopy of stool, and this is very insensitive. Now with the molecular diagnostics available, it has increased sensitivity, but still there is no good gold standard because of the fact that most people are chronically infected with low and intermittent larva output. Therefore, serology must play an important role to help in diagnosis. However, the serological tests available in the market are all IgG ELISA, and there is no commercial rapid test available at this moment. Next. So what we have developed are 3 tests. The main one that I'm talking about is the IgG4 lateral flow rapid test, which we call SS rapid. We also have developed an IgE ELISA using a novel strongyloides recombinant protein, and an antigen detection ELISA using a novel SCFV recombinant monoclonal antibody. Next. So first I will talk about the SS rapid. Like I said, it's a lateral flow rapid test. First, we developed it in the form of a dipstick because in this format, the resources are less and we only need 10 microliters serum. And serum samples for this kind of investigation is very precious, not easy to get. And after we have developed it to a good level and we are happy, then we convert it to the cassette test. As you can see, it requires more than 3 times sample, 35 microliter. It is an IgG4 detection. Why did we chose IgG4? Because this is a chronic infection and a helminth infection whereby IgG4 levels are high. Yeah, and also this isotype usually is more specific when used in an assay. So the test line is the test line in the arrow is the recombinant NIE, and the top control line is anti-mouse IgG. And the SES page shows the recombinant protein, which is quite pure. The little band at the top is also recombinant NIE, it's just a little clustered protein over there. Next slide. Okay, so we have done many studies on it. So the first dipstick study, we did an evaluation of the sensitivity and specificity. We have published in American Journal of Tropical Medicine and Hygiene, and we find it to be 91% sensitive and 100% specific. And in another university in Malaysia, we sent the test and they also found it to be highly specific. We also sent to Italy, where they did a preliminary testing with about 10 microscopy positive people, and it was all positive, and the specificity is also good. We also tested in Iran with a high sensitivity, and we also sent our rapid test to Thailand, and over there they found the sensitivity slightly lower, 82%, but the specificity is high, and this result has been published. We have used the dipstick also to test immunocompromised patients at one of the hospitals, 200 patients, and we find that the prevalence is 10%. Okay, next. So so we also did a study on serum absorption to validate the specificity of the rapid test, because there are some samples, we have to prove that some samples which apparently seems to be false positive, but actually were indeed real positive by absorbing, by making beads, by putting the proteins on the beads, polysaccharide beads, and absorbing the serum. And then if it is a real strongyloides protein, after absorption, the test becomes negative. And then if we absorb it with the beads that is coated with a different recombinant protein, there is no change in the test result after the absorption. So if you'd like to know more about it, you can look up in this published paper. Okay, next. So after that, we converted it to a cassette test, and this is how it looks like. We can grade it into different intensity, +1 to +4, and the negative result has no test line, and the bottom is the configuration of the cassette test. Next. So this test now has only 3 simple steps, the procedure. So it's the point of care test. It's field applicable, and it takes about 15 minutes if you use serum or plasma, and about 20 to 25 minutes with whole blood, because it takes time to clear the blood, the red background. Okay, next. We also tested it with, we also showed that if you take a positive serum and then you take a blood that is spiked with the same serum, the intensity of the test is the same. So we tested it with at least 10 different samples, and we showed that the test works well with both serum and blood. Okay, next. So the cassette format, we have just published it in Acta Tropica, just out, and this is the first evaluation of the cassette format. Okay, next. So these are some of the studies that have been done or are being done on the cassette format. So in the paper I just showed you, the sensitivity was tested with samples from Malaysia, Iran, and Thailand, and also the specificity is 95%. And all of those which are false positive, we did serum absorption, and they become negative after the absorption. We also tested on cancer patients which are treated with corticosteroids, and we find that there is about 11% prevalence. Other than that, we have another person in another hospital that tested the category 4 and 5 of the COVID-19 patients, because as you know, these patients are given steroids, and there are 14% prevalence among them. And currently there are 2 studies ongoing. One is a WHO field study in Ecuador, and also another retrospective study in the WHO Collaborating Center in Italy. Okay, next. Just to show you, this is a comparison of the SS Rapid versus the IgG ELISA using two commercial tests from Euroimmune and Bodier. You can see that SS Rapid only did not detect 1, while the other 2 ELISA did not detect more than 1. So, but in general, there is good concordance among the tests. Okay, next. So the next assay that we developed is an IgE ELISA using a novel recombinant protein, and IgE because we are trying to find an assay that may replace IgG, because IgG tends to have less specificity. So we have published this paper in Diagnostics. Okay, next. So in the evaluation, we divided the samples into 4 categories. The first one is where the patient's stool is either microscopy positive or PCR positive, and the serology is of the serum is positive, and we think these are probably chronic infection. And another smaller group where the microscopy is negative, but the PCR is positive and the serology is negative. This is very interesting. So because the serology is negative and the PCR is positive, we think this is probably early acute infection where the levels of the antibody has not risen to a detectable level. We have two groups of control infections. One is other infections and healthy donors. These are from which were tested negative by the serology, and so these are the control groups. Okay, next. So we find that this has high sensitivity and specificity, with a cutoff value of 0.22. Next, and a good, of course, area under the curve. So here it shows that the different groups are having the average OD it's significantly different between positive and negative. And interestingly, the chronically infected group has got a higher average OD than the acutely infected group. Okay, next. These are the distribution of the OD values. So it's clearly differentiating positive and negative. Okay, next is our antigen detection ELISA. This has also been published in Acta Tropica this year. Okay, next. So here we also use the same for serum sample, but only the numbers are a little bit reduced because by that time we finished some of our samples. And next, here also the ROC curve. Next, please. ROC curve also shows very good sensitivity and specificity. Next, please. So here you can see that the OD values, the average OD values are also mostly significantly different between the different positive and negative groups. Can you click on to the next one? Please click. So I bring up the IgE ELISA graph to show you that, so in an antigen detection ELISA, we can see that the probably acute infected patients have higher OD than the chronic patient versus the IgE ELISA where the opposite we was seeing. So this is rather interesting, but we have to reserve this finding for more investigation because the number of samples are still not high, but it's something that we should further investigate. Okay, next. So in conclusion, the SS rapid test, we feel that it showed good diagnostic value because we have done quite a number of studies, and it's already a prototype test. And we like if other people are interested to evaluate it further, and also to include some post-treatment studies to see how long after treatment the test becomes negative. And as for the IgE ELISA and antigen ELISA, it has good potential, and definitely more evaluation studies are needed. Okay, next. I'd like to acknowledge, next slide. These are some of the other contributors in this project and the research grants that funded them. Okay, thank you very much. Next, please. So the last slide is a thank you slide. Thank you, and I'll be happy to answer any questions if you have.